living on the bottom. Macro, that word macro, means the critter's big enough that you can see it without the aid of a magnifying device, like a microscope or something. And invertebrates are animals without backbones, ranging from jellyfish to sea urchins or starfish, sea stars. Some of the gear we use to sample macroinvertebrates, and I guess, Ashley, I got to ask, is everybody just seeing the presentation? Because I'm getting lots of other stuff on my screen.
I can see, yeah, just the presentation. Yep.
Okay, very good. That's what we want. So some of the gear that we use to sample these guys, various kinds of dredges or grabs, like on the left there, that's me a long time ago on the Santa Fe River when I worked for the Swanee River Water Management District with a Ponar grab. On the right, various kinds of nets. That's a dip net in my hand, kick and seine nets. We can use various kinds of coring devices, or in streams, these things called Serber and Hess samplers, or what we call artificial substrates, something that you hang in the water and leave it there for generally anywhere from 2 to 4 weeks. And the invertebrates collect and colonize on this thing. So those are some of the gear types that we use to sample benthic macroinvertebrates. And they have been used for a long time in what we call bioassessment. That word refers to measuring or monitoring something biological, whether it's plants or plankton or birds or benthic macroinvertebrates, as a kind of an indicator of environmental quality or environmental health. And benthic macroinvertebrates have been used for decades in the bioassessment of water quality. And I'll come to that in a little bit. Some of the reasons why they're good for that are they're sessile. They're either fixed in one place or they have limited mobility. They can't move around really far. And combined with the fact that they can be sensitive to water quality changes. So if water quality degrades or gets worse, things are going to happen to the benthic macroinvertebrate community. A lot of them have fairly specific habitat requirements, so they're also affected by physical things like dredging and such. And that the opposite of that would be fish, which maybe are not as good for bioassessment because they're much more mobile and they can avoid nasty conditions. So as I mentioned, macroinvertebrates have been used extensively in the bioassessment of water quality and freshwater ecosystems for decades, probably almost for the last century, going back to the early 1900s. Actually, back then, the chemistry was pretty rudimentary. So a lot of the things we can measure today to measure water quality, they couldn't do that back then, but they actually were able to assess water quality by the composition of the benthic macroinvertebrate communities. It was this scheme developed mostly by European biologists called the saprobic system. Nowadays, with the Clean Water Act and other regulatory tools, a lot of the things they had back then, the big nasty sewage treatment plant discharges and the industrial discharges, that's been cleaned up due to regulations and other things. And nowadays, one of our major sources of water quality, water pollution, what we call these non-point sources, things like dispersed sources like septic tanks, stormwater runoff from urban land uses or agricultural land uses. And these are kind of, they don't occur all the time. They occur in short-term pulses that can be very difficult to measure with conventional water chemistry sampling, but the benthic invertebrates tell you what's going on. And we can use them to assess other impacts in freshwater ecosystems as well, like changes in water flow or changes in habitat. They've been also used some in coastal waters, estuaries and coastal waters, but not as extensively as in freshwater because a complicating factor there is salinity, both natural changes and human-caused changes in salinity. So benthic macroinvertebrates are a major group, I would even dare say probably the major group of animals found in freshwater ecosystems. They're found in rivers and streams. That's the Econ River there on the left. They're found in the bottom of lakes and ponds. That's Lake Poinsett. And they're found in freshwater wetlands, and I believe that's Payne's Prairie south of Gainesville. But they occur in all of these freshwater ecosystems. And why are invertebrates important? Benthic macroinvertebrates important. What are their ecological roles? Number one, they are important, what we call primary consumers. They're vegetarians. They feed on either live or dead plant material. And in so doing, they convert that to their bodies, to animal biomass. They are a very large component of the overall animal or faunal biodiversity. In fact, for a given aquatic ecosystem like the Econ River or Lake Poinsett, if you were to sample either of those thoroughly, you're going to get more species or taxa or more different kinds of benthic macroinvertebrates than fish, birds, reptiles, amphibians, and mammals combined. So they're a large element of that, what we call biodiversity. Some benthic macroinvertebrates are key intermediate predators, and some are important algal grazers, helping control algal proliferation. And perhaps most importantly, they are the food for things that we worry about or are more interested in, like fish. A lot of fish feed on benthic macroinvertebrates, birds, and other animals higher up on the food web or the food chain. Some of the major groups of benthic macroinvertebrates in freshwater ecosystems are snails and clams, the mollusks, a lot of different species of oligocete worms, various crustaceans. Probably the one everybody's familiar with, or the fourth one there, the crayfish or crawdad. But there are some other freshwater crustaceans, a few species of shrimp, and some little teensy things that I'm going to show you in a bit called scuds and sowbugs. Probably the major in terms of the numbers of different taxa or kinds of benthic invertebrates in freshwater ecosystems are going to be the aquatic insects, various kinds of aquatic insects. And when we talk about these guys, we have to recognize the two major types of insect life cycles, and I'll show you why that's important in a bit. On the left, a lot of insects have what we call this complete metamorphosis life cycle, where the egg hatches into a young stage called a larva. And then before that larva turns into an adult, it goes through a pupal phase. It turns into a pupa or a cocoon. That's on the right. Now on the left, there are many other groups of insects where the egg hatches into a young phase that we call a nymph. And you'll notice instead of going through a pupal phase, the final molt of the nymph turns into the adult. That's called incomplete metamorphosis. Reason I point this out is for a lot of aquatic insects, that's what we're collecting. We're collecting the larvae or the nymphs, the young phase. The adults are terrestrial. They fly around on land, but it's the young phase that lives in the water. That's aquatic. When we talk about aquatic insects, some major groups of aquatic insects are entirely aquatic, meaning all of the members of that group, all of the species, have to live their young phase in the water. They need water to complete their life cycle. So mayflies, stoneflies, caddisflies, and your odonates, your dragonflies and damselflies. These are entirely aquatic. Other insect groups on the right there, the list on the right, may have aquatic subgroups. So the beetles, probably the largest single group of animals in the world. Half of all known insects are beetles. Many beetle groups are entirely terrestrial. They live their entire life on land, but then some groups of beetles are aquatic, like the family Dytiscidae, the diving aquatic beetles. And then some of those are entirely aquatic in that you find all life stages in the aquatic environment. So their eggs, their larvae, and their adults are aquatic. And that's my little note down there. Mainly some of the beetles and some of the true bugs or hemipterans live their entire life up through adults as in the water as aquatic. So to introduce you to some of the major groups of these benthic macroinvertebrates that we do get in the Wekiva River. I'm going to introduce some of those, talk about some of those, and then also how we use these to assess water quality and bioassessment of water quality. Basically, we kind of break these guys into three groups: Group 1, Group 2, and Group 3. The Group 1 are the pollution sensitive groups, so very sensitive to any kind of water pollution or degradation of water quality. And probably the top three are the mayflies, which you can tell those guys, these are the nymphs, the young phase. They have three filamentous or thread-like tails. In terms of their life habits, they're kind of crawlers, clingers, like that guy in the picture there, or some of them are pretty good swimmers. They swim around. So that's the one group, and we do get maybe 12, 15, almost 20 species of mayflies in the Wekiva River. Second group that we're going to talk about are the stoneflies. Again, these are what we collect in the water are the nymphs. They have two filamentous tails. They're mainly crawlers. They live on hard surfaces like rocks and logs and such. We don't get many of those guys here in Central Florida. Historically, there was probably maybe one taxon of stoneflies that we found in the Wekiva River in the genus Neoperla. These guys like cooler water and water that they only live in rivers here in Florida, so very, very good flow all of the time. So that's mainly we find these guys in North Florida streams, but historically there was maybe one or two in the Wekiva River. And then the third group are the caddisflies. They're in this case, these are larvae. They're kind of caterpillar-like in appearance because these guys are closely related to the Lepidoptera, the butterflies and moths. And many of the caddisfly larvae construct some type of a case. Some of the little guys, the purse caddisflies, actually use individual sand grains to build a little case around them. And the bigger guys use pieces of leaves or twigs to build a case. These three groups, the mayflies, stoneflies, and caddisflies, are three most pollution sensitive groups. They constitute what we call the EPT taxa, which are the scientific names of these three groups. And in a lot of states, including Florida, that use these guys, they develop some kind of an EPT index, a number, a quantitative measure based on the abundance or occurrence of taxa of these guys. Some of the other group 1 taxa that we get in the Wekiva River and here in Florida, the Dobson flies, it's a fairly small group of insects, but the one we get in the Wekiva, Corydalis, they produce a larva called a helgramite. And I mean, think of this thing as a caterpillar, not on steroids, but on radiation, like the Incredible Hulk. You can see they get pretty big, pretty fierce looking. They do bite, they will draw blood, but they're very sensitive. They need good, clean, well oxygenated water. Generally find them on and under hard substrates like rocks and logs. So they are a good water quality indicator. If you're canoeing, kayaking down the river and you see a branch hanging over the water and what looks like bird poop on the leaves, white dribblings, and you say, "Ooh, bird poop." But if you go up to it, you'll actually see it's not bird poop. It looks like something actually glued these things to the leaves, and those are the egg cases of the Dobson fly. And then some of the mollusks in the Wekiva River, some of the gill-breathing snails, particularly that guy on the right there, that is the Florida Alemia or Florida riverhorn snail. We do get those on the Wekiva. And some of the stream mussels in the Wekiva are fairly sensitive to, so they're a group 1. The group 2 taxa are what we call moderately pollution sensitive. They can tolerate a little bit of water quality degradation, but generally still need a good water quality. And one of the major groups here are the odonates, which are your dragonflies and your damselflies. Again, these are the young, the nymphs. Your dragonfly nymphs are generally stout-bodied. Some can get quite big, a couple inches long. They have internal gills. In terms of life habits, they mostly crawl about on the bottom or in vegetation or sprawl on the bottom, or some of them are burrowers. Those two pictures there, the one on the left is a dragon hunter nymph. That's a big dragonfly. We get those on the Wekiva. And then in the spoon there, that's a clubtail nymph. Most of those burrow into the sand or the mud. They create a little shallow burrow. And then down below, the damselflies, their nymphs are generally smaller, more slender bodied. The thing about these is they have the three paddle-like tails or gills. Remember, mayflies, the tails were thread-like or filamentous, whereas the damselfly nymphs have these paddle-like tails. Those are their gills, and they're largely crawlers, clingers, like you see in those two pictures there. Some of the aquatic beetle larvae are in the group 2, moderately pollution sensitive. Some of the diving beetle or diticid larvae, and some of the whirligig beetle larvae. The whirligig beetles are those little guys, you'll see them sort of skating around on the top of the water, and they occur in fairly moderately sized to large groups, all sort of clustered together. Those are the gyrenids, but their larvae are can be fairly somewhat sensitive. So group 2. A lot of your aquatic crustaceans are in group 2. In particular, your crayfish or crawdads. I think everybody knows those. Think of those as a mini freshwater lobster. They get up to a few inches long. And then the grass shrimp, we get a couple species, at least one I know, in the Wekiva River. They don't get as big, mainly up to maybe about 1 inch long, a very clear body, long, slender antennae. And they have this thing between their eyes. I call it a Klingon dagger. Think of the Klingon knives, you know, that have all those scalloped edges. It's called a rostrum, but it's between their eye stalks. And then a couple of the other freshwater crustacean groups, these guys, very tiny. They don't even get to be 1/2 an inch long, usually only about 1/4 of an inch, a centimeter or so in length. And those would be the amphipods or scuds. We get several species in the Wekiva River. They kind of scud or scoot about in your tray. And then the sow bugs, the isopods, we get a couple species in the Wekiva. They resemble the pill bugs or the roly-polies, which are actually terrestrial isopods. And so they're kind of top to bottom or dorsoventrally flattened. So those are group 2. And then, okay, and then the third group is the group 3, the pollution tolerant groups. These include some of the chironomid midges, the red wigglers, their larvae. In this case, we're talking larvae because they're in the flies group, flies, midges, mosquitoes, that kind of thing. Again, they don't get very big, you know, barely 1/2 an inch long or so. Some of these guys are a bright red. You know, if you pull up some dirt in your anchor and you see, like I say, these little red wigglers in the dirt, those are probably chironomid midges. A fair number of your oligochaete worms are fairly pollution tolerant. And then a lot of your air breathing groups, these are mostly the adults of some of your aquatic hemipterans and your beetles. That's a diving beetle there on the left, the picture, an aquatic beetle. And on the right there, that's a water scorpion. That's a hemipteran. And you can see that thing on his back is an air tube, like a snorkel. So he's breathing through his spiracles, but he's got that thing poking above. So a lot of these guys are tolerant of pretty nasty water. Now, because you see these guys, they don't in and of themselves indicate pollution. It's just the fact that they have broad environmental tolerance. So in a polluted type situation, you're going to lose all of your group 1, a lot of or all of your group 2, and these are going to be the only guys you find. And in fact, that's what I kind of show you here. As aquatic scientists, we've developed these concepts of a biological condition gradient with this graph. So on the bottom there is some type of a stressor. In this case, it would be some kind of a water pollutant going from low to high concentration. And then so that's on the bottom. On the left of the graph would be some type of a numeric reflection of the biological condition. So at the top left there, that big number 1, that's going to be what we call the natural or reference condition, where you've got pretty much a completely natural benthic invertebrate community. You've got lots of group 1 taxa, you've got lots of group 2 taxa, and you'll have some group 3 too. Now, as you begin to get some water quality degradation, you sort of move down that stressor gradient, you move to that big number 2 or 3. You start to lose some of your really sensitive species, like some of your stoneflies and such. Maybe the abundance of some of your group 3 taxa starts to increase in your sample. Little more water pollution and you get to the 3 or 4, you pretty much lose all your group 1. You maybe have some group 2 taxa, and you're really starting to mostly collect in your sample those group 3 taxa. And when water quality gets really bad, pretty much you lose all of your group 1 and group 2, and only find the group 3 taxa. So that's kind of how we use these guys to as to do bioassessment of water quality. In Florida, they've developed several tools, bioassessment tools, to sample macroinvertebrates and then to take them back to the lab and to calculate some numeric measures that I'll show you a list in a little bit. They developed back in about 2000 or so, the late 90s, this thing called a stream condition index. And then later on, they developed a little shorter, quicker version called the biorecon, or biological reconnaissance. But this involves going out, sampling the invertebrates, and then looking at what you get. And this was mainly done by our state Department of Environmental Protection, and they've got a really interesting website. The URL is down there that you can go to, and they've got some of it's pretty, it's for scientists, but they've got some really good descriptions of how they use this stuff. Some years ago, they also developed a lake condition index using macroinvertebrates, but over the past years, they've statewide, it does not seem to be as valuable in indicating a lake condition using the macroinvertebrates. But some of our counties still use it. Seminole County still uses the LCI when they go out and they sample macroinvertebrates in some of the Seminole County lakes. It does a very good job of telling them the water quality of the lake. They get a very good LCI in a clean lake, and not so good in the more polluted lakes. So the stream condition index, I'll just mention that, is what we call a multi-metric index. It's actually made up of these 10 things here. So these are the things that you get from your macroinvertebrate sample. The total number of different kinds of invertebrates, we call that total number of taxa. And then we also know some of these guys, like some of the odonate larvae and that helgramite, they take a long time to finish their life cycle. So these are those long-lived taxa. So the number of those, number of mayfly taxa, number of caddisfly taxa, the number of Florida sensitive taxa. There's an old index called the Florida index, and then others. And all of these are combined to form this single number called the stream condition index. It was actually sort of the idea for this kind of a thing came from the economic indices we use, like the Dow Jones Industrial Index or the NASDAQ. It's one number, but when the Dow Jones is down, your mutual fund or your stock portfolio isn't doing very well. When it's up, you're making money. And so it's the same kind of a deal, this single number that gives you this overall assessment of the condition of the water body. You can see down at the bottom there, an SCI score of 100 to 64 indicates exceptional. So that would be a reference or background condition. 63 to 40 means still a healthy water body, but 39 and below means some kind of a water quality problem, which we call impaired. So that's a whirlwind introduction to the benthic macroinvertebrates of the Wekiva River and how we use them in the assessment of water quality. I surely do wish we could be down at Katie's Landing. That's where we were supposed to be today. And I would have been there with my dip nets and a table and trays and aquaria and such. And we would have been looking at some of these guys live, and I would have been telling you about them. But so we had to, times being what they were, had to go to the virtual. But the last thing I want to show you, this is another guy from the Wekiva River. This is the giant river prawn. The genus is Macrobrachium. This is probably Macrobrachium carcinus. A couple years ago, Deborah Shelley, who's going to be talking to you in a couple of weeks, she's our retired aquatic preserve manager, and Larry Spooner, who lives on the lower Wekiva, Larry put out some minnow traps one evening off his dock. And when he came out the next morning and pulled them, this guy was in one of them. And so he sent these pictures to Deborah, and Deborah would always send pictures to me with like, Rob, what is this? Because, you know, nobody had ever seen this before. And you rarely do see them. They only come out at night to forage. They are a shrimp. They're a freshwater shrimp. This particular guy is native to Florida. He does occur, he's been known to occur in the St. Johns River and its tributaries. But so that's the giant river prawn. You get a little better view. Again, he is a shrimp. So remember I said the shrimp. So there you can kind of see the Klingon dagger there between his eyes. But this is yet another one of our Wekiva River benthic macroinvertebrates. And that's all I've got to talk to you about. If we've got some questions, I'd be happy to answer them.
Well, thank you, Rob. That was great. I always enjoy your presentations. I do have one question. What is your favorite macroinvertebrate?
My favorite macroinvertebrate. Yeah, I thought that'd be coming. I suppose my favorite group are the stoneflies. You know, back when I worked up in North Florida, we collected a number of stoneflies in the Swanee when I worked in the Swanee, and and then they indicated, you know, good water quality. We actually collected some that nobody had known that they actually extended out of the Panhandle into North Central Florida. But so a few years ago, when I turned 40, I wondered, you know, what can I do to show my commitment to clean, healthy rivers? And then this little place opened up in downtown Lake City, Pete and Renee Tattoo and Body Piercing Emporium. So I worked up an illustration of a stonefly and I brought it in and asked Renee, can you guys do this on me? And they said, yeah. So I've got a stonefly nymph tattooed on my left arm, and that shows my commitment to clean, healthy rivers. Although I got to admit, I'm partial to this big guy here too. He's pretty cool.
Yeah, I agree. All very interesting, and I always enjoy finding them. Again, thank you so much.